Good morning, everyone. Thank you for having me, and many thanks to the ISEC organizers for the invitation to give this plenary. Today, I'm going to tell you a bit about what we're doing on large carnivores in our team and how statistics may help to address some questions we have about their ecology and their management. Because I cannot be with you in person, let me start by introducing myself. My name is Olivier Jimenez, and I spend quite a bit of time on Zoom, like many of you, I assume. I like fishing, even though I'm not really good at it. And I work somewhere in France, in a beautiful city on the south coast of France called Montpellier. And Montpellier has many charms, which I promise are not the reason why I'm not in Cape Town this week. I really... Okay, I work in a research center called the Center for Functional and Evolutionary Ecology, which looked like that in the 60s, some sort of star with three branches. And this is what our building looks like today. It hasn't changed much. It just got a bit busier around. Our research center has amazing facilities like convenient places to park your bikes, like here, and a pioneering architecture. In terms of background, while I struggled with studying math, obtained my PhD in applied statistics a long time ago in a galaxy of wine and cheese far away from South Africa. And I obtained my habilitation in ecology and evolution so that I could stop pretending to understand what my colleagues were talking about. And more recently, I embarked in sociology studies because, hey, why not? Okay, so before I tell you about our work, let me give you a bit of context about how I ended up giving this talk, how it all started. Okay, I received an email from David Wharton and Rez Adveg invited me to be a plenary speaker. I was really on cloud nine because ISEC is probably my favorite conference. That was back in March 2021. Well, I was quite relaxed at that time. And then in February 2022, my Twitter feed got crazy with folks sharing the acceptance of that talk. I felt some discomfort on my scale of stress and anxiety. And then 3 months ago, I got an email from Caroline Melnick giving the good news that I should record my talk at least a week prior to the beginning of the conference. I could feel the stress rising up, but 3 months is an eternity in the life of an academic, I guess. So, hey. And then the video recording session was set up for me in my agenda. How thoughtful. This kind of triggered something in me. And maximum level of stress was reached when everybody started sharing the topic of the talk on Twitter. And what do I do to reduce my stress level? Well, maximum procrastination. I watch a ton of TV series. I mean, guys, the world is against me. Obi-Wan Kenobi, Star Trek, right when I'm supposed to work on my plenary seriously. Fortunately, the ISEC organizers were very supportive. Okay, what have I learned from this very productive period? Well, first, I spend way too much time on Twitter. Second, the ISEC organizers are doing a great job. Thank you very much to them and to the organizing and the scientific committees. And sadly, getting old does not mean that you manage your time better. On the contrary, actually. And again, I spend way too much time on Twitter. Okay, let's get to the topic of my presentation, starting with statistical ecology. To begin with, I'd like to give you some definitions to set the scene. First, what is ecology? Well, if I talk to my mom, she thinks that I'm traveling in amazing places, just wandering around, looking for beautiful animals. But what ecologists do is a bit less glamorous. Ecologists do fieldwork. We collect data to test ecological theories. We also do lab work, again, to collect data and test ecological theories. And we also attend boring meetings, get tired and fall asleep. In brief, ecology is the study of how organisms interact with one another and with their non-living environments. Now, what is statistics? Well, what my mother thinks I've been doing all these years is playing with a bunch of computers, and that my desk looks like a scene from the Matrix movie. Not to say that my mother has seen the Matrix movie, but I don't think she has. A question that is often asked is whether statistics is the art of turning correlation into causation. In reality, what statisticians do is trying to make sense of data and uncertainty with some sort of formalization, often using equations. We spend long hours debugging code and running models. And like ecologists, statisticians attend boring meetings, get tired, and fall asleep. Statistics is usually defined as the science of collecting, organizing, presenting, analyzing, and drawing conclusions from data. And I insist on the collecting, organizing, and presenting steps. Statistics is not only about analyzing and drawing conclusions, far from it. Now, the question I'd like to raise is, can statistics and ecology make a happy wedding? Well, I don't need to convince you, I guess. You wouldn't be attending ISEC otherwise. But let me tell you how we combine statistics and ecology in our team to contribute to the conservation of biodiversity and large carnivores in particular. So we have 4 species of large carnivores in Europe: wolves, bears, lynx, and wolverines. And today I'm going to talk about wolves and lynx mostly. Globally, large carnivores historically covered almost all of the world's land area. And now, intact large carnivore guilds, those with 0 carnivores lost, occupy only a third of the world's land, which you can see in orange on this map. What is the situation in Europe? Well, the 4 species of large carnivores are spread over more than 40 populations. And their main characteristics is that several of these populations are transboundary. That is, they live across several countries. All these species were once at the brink of extinction, and they have recovered in the 20th century. This recovery is often used as an example of a success story in conservation biology. What are the reasons for this recovery? Well, these reasons are, among others, agricultural abandonment, with the comeback of ungulates, the main prey of large carnivores, and large carnivore species have also benefited from laws on nature protection. This recovery, the recovery of large carnivores, also means more interactions with humans. Here you have Here you have a lynx near a big city in Switzerland, a wolf looking for food in Germany, or a bear here wandering around near a village in Italy. These interactions come at a cost for humans with depredations on beehives or livestock, and conflicts between groups of people too. Here a group demonstrating and arguing that pastoral activities are not compatible with the presence of large carnivores, and here another group demonstrating and saying no to the hunting of wolves. How do we contribute as statistical ecologists to a better understanding of these conflicts? Well, by going back to questions, and pretty basic questions actually. How many? How do we count large carnivores? And where? How do we know where they are? And how to address these questions? Well, we're going to see that a class of models called hidden Markov models are particularly useful to do that. The problem is that detecting animals, individuals, or species in their natural habitat is difficult. Difficult due to weather conditions, for example. Difficult when species habitat is not open. Also, animals do not make our life easy, and their behavior can make it difficult to see them. Of course, observing is also a matter of skills, with some observers being less dedicated to the task than others. Overall, what you and what we see in the field might not be exactly what there is, and we don't have the full picture of what is happening. This is due to the issue of imperfect detection. The probability of detecting an individual or a species is less than 1, and not necessarily the same for all individuals in time or space. And nowadays, to address this issue of imperfect detection, we use more and more hidden Markov models. Briefly speaking, these models allow you to distinguish the ecological states you're after from their observation. By true state, I mean alive or dead for an individual, or present or absent for a species at the site. It is said that hidden Markov models were initially developed to guide satellites, where the ecological process would be the true position of the satellite, and the observation are coordinates that are prone to measurement error. Okay, now we don't have nothing as fancy as satellites for wildlife, or do we? We observe animals on the field, and we'd like to infer from the data some ecological processes or mechanisms possibly explaining what we see, right? What are these hidden Markov models? Okay, imagine Tom and Meg are in a long-distance relationship. Imagine it's sunny outside Tom's house. And when Tom and Meg talk on Zoom, if Tom tells Meg that he's happy, then Meg can infer that it's sunny outside Tom's house, okay? Just from Tom's mood. Whereas if Tom is grumpy because it's raining outside, well, Meg can infer that it's raining just by knowing Tom's mood, right? The hidden state here is the weather, since Meg cannot see the weather, and she can only make inference from Tom's mood. Of course, things are a little more complicated than that, and if Meg could see the weather perfectly, she would realize that there is some stochasticity. And if it's sunny, it's a sunny day, there is an 80% chance that the following day will also be sunny. And therefore, there is a 20% chance that the following day will be raining. Oops. When it's raining, there is a 60% chance the following day will also be raining, and a 40% chance that the following day will be sunny. But Meg cannot see the weather, and she has only access to Tom's mood, happy or grumpy. And given the weather, there are some probabilities that Tom is happy or grumpy. All of these probabilities are from previous data that has been studied by let's imagine some data scientists at Zoom. And with these probabilities, Meg can infer the weather if Tom tells her his mood was, for example, happy, grumpy, grumpy, throughout the week, right? What can we do with HMMs? Well, you might want to estimate the probability of transitions between the hidden states, between rainy and sunny states. You might want to estimate the observation parameters, that is the chances of being happy or grumpy given the weather. To do so, you can write down the likelihood and get parameters posterior distribution with Bayes and MCMC methods. Alternatively, you can marginalize the likelihood with the so-called forward algorithm and get maximum likelihood estimates, or MLEs, for all parameters. You may also want to infer the hidden states themselves, not just the transition probabilities between these states. Again, you can go either directly with Bayes and MCMC methods, or you can decode the hidden states with the so-called Viterbi algorithm. Okay, now that we have our modeling framework, let's get back to our initial motivation and the ecological questions. Let's start with the how many question. Counting things sounds easy, but is it really specifically, can we count animals exactly? Well, we know that it's not an easy task, at least since the 18th century and the first application of capture-recapture by Laplace. Laplace proposed an estimate of the population size of France by using two samples, one from the churches and their birth registries, and another one through a census of the population. In ecology, we did with this kind of material, scats. Why scats? Well, because large carnivores are like sheep machines. These animals are kind enough to leave all sorts of signs of their presence in the field, like this brown bear here in Greece, who is rubbing on a pole with barbed wire, leaving hair for us to analyze. We then use the DNA contained in scats or hairs to infer the identity of species or even individuals through sequencing. And in our team, Lucille Maresco on the left and Sarah Cubain on the right have worked a lot with this data during their PhD and postdocs. How do we model capture-recapture data with HMM? Well, for each individual, you may be alive or dead at each sampling occasion. These are our weather states in the long-distance relationship example. Of course, to be first captured, you need to be alive with probability 1, right? Then comes our Markov model for the dynamics of the states. Your state at t+1 only depends on your state at t. Say you're alive at t, then you may be alive at t+1 with probability phi of survival, or dead with probability 1 minus phi. In contrast, if you're dead at t, then you remain dead at t+1, okay, with probability 1. So these two blocks define what we call a Markov model, but the issue of imperfect detection makes it a bit more complex, and we need a third block to describe how these states are actually observed. And this is the observation process here. If you're alive, then you're detected 1 with probability p and missed with probability 1 minus p, the detection probability. Now, if you're dead, you're not detected with probability 1, right? And this is all good, except that parameters are all homogeneous. But nature likes variability, and this is the raison d'être of statistics. So what about individual heterogeneity? Well, from a statistical point of view, heterogeneity can induce bias in parameter estimates. And from an ecological point of view, heterogeneity is of importance, and you might be interested in studying individual quality or individual behavior, for example. How to account for heterogeneity? Well, if variability is observed and measured in some way, you may use this information in your models with individual covariates, for example, like body mass, or group effects, like sex. If not, well, you can use random effects or finite mixture models. What are the sources of heterogeneity? Well, in wolves, for example, there is a social hierarchy with dominant versus subordinate individuals. And dominant individuals are more mobile within pack territory and more likely to mark territory with feces and urine. And Charlie Pledger, back in the 90s, developed models to account for heterogeneity based on finite mixtures. In brief, individuals are assigned to a class or another with some probability, and survival and detection parameters can be class-specific. So how to write this model with HMMs? Well, individuals can be in state alive in class 1 or in class 2 or dead with probability pi and 1 minus pi, right? The transition matrix is extended with an extra row and an extra column to account for both alive states. Here, survival is not class-specific, but it could be. And also note that if you're alive in class 1, you cannot move to class 2. You stay in the same class, right? This assumption can be relaxed by introducing two new parameters, psi12 and psi21, which are transitions among classes. Ecologically speaking, think again of wolves, subordinate individuals can become dominant, for example. Okay. Let's get to the observation process. Now, in contrast to survival, we make detection class-specific. Here, P1 and P2 for detection in class 1 and class 2. And if you're alive in class 1, then you may be detected with probability P1 and missed with probability 1 minus P1. And same thing for individuals alive in class 2. Using these models, Sarah and Lucille have produced estimates of abundance for wolves abundance on the y-axis, over the years on the x-axis. Here you see that the raw counts, here the plus signs, are not the whole story. And once we correct for imperfect detection, the estimates, the solid line here, are higher than what we see, of course. And we see that the wolf abundance has increased over time, and we have between 600 and 900 wolves today in France, if we believe our model, of course. An important thing to note is that these estimates come with a measure of uncertainty that is sometimes difficult to convey to stakeholders. Note that if we ignore heterogeneity in detection, the dashed line here, there is a 30% underestimation of abundance, which has obvious management implications. Another issue related to the management of carnivores is the existence of hybrids and how many there are. Not these hybrids, but those ones, hybrids between wolves and dogs. And more specifically, we study anthropogenic hybridization, which is the interbreeding between individuals from genetically distinct populations due to human causes, human action. And anthropogenic hybridization is considered an important and increasing threat to biodiversity. Why? Well, because the domestication process has been associated with intensive selection for traits that may be a disadvantage in the wild. Okay. So a very good example of that is the hybridization that occurs between wolves and dogs at increasing rates, at least in some areas in Europe. And this phenomenon is raising concern in the scientific community to the point that it's considered one of the main threats to wolf conservation in some parts of Europe. Surprisingly, we know very little about anthropogenic hybridization, and we have no estimates of the proportion of hybrids that are present in wild populations. It's a quantity that we call prevalence of hybrids from now on. So far, studies have been focusing on naive prevalence, the proportion of hybrids in the sample, but we know that it's not a good estimator of the population at the population level because detectability has to be considered. And plus, there is also uncertainty in the state hybrid. Uncertainty is due here to the fact that hybrid identification is performed on DNA extracted from wolf scats, and for some individuals, the classification is not clear. So Nina Santostasi worked during her PhD on the topic. She developed an HMM to estimate the proportion of hybrid. So you start with the three states, alive as a wolf, alive as a hybrid, or dead, with proportion pi W and 1 minus pi W. There is a state-specific survival, different for wolves and hybrids, of course. And what data do you collect on the field? Basically, you have 4 types of observations. First, an animal may go undetected. We code that 0. Or detected as a wolf, which we code 1, or a hybrid, which we code 2. And last, an animal may be detected, but we're unable to assign it a state, hybrid or wolf, for the reasons I explained before, and it's coded 3. Okay, now the observation process. It can be summarized as follows. If you're alive and wolf, then you may be undetected with probability 1 minus PW, the detection probability of wolves. Detected as wolf with probability, the detection probability, times the probability of being correctly assigned as a wolf, denoted delta W here. You cannot be detected as a hybrid, and you may be detected as uncertain, the 3, with probability, the probability of detection times the probability of being not assigned as a wolf. And you proceed the same way for individuals alive in the hybrid state. And of course, if you're dead, then you're undetected with probability 1. This observation process can be split into 2 sub-processes. First, you have detection in this matrix here. And then, conditional on detection, you have the assignment process. And the full matrix we saw just before is the product of these 2 matrices. And Nina, using this model, fitted it to a data set collected in a protected area in Italy that hosts the territories of about 7 known packs over 5 sampling occasions. She found that survival was higher for hybrids. And then she calculated naive prevalence by mimicking the usual practice of assigning uncertain animals to wolves. Okay? And using the Viterbi algorithm and the Horvitz-Thompson method, Nina estimated prevalence by accounting for imperfect detection as well as uncertainty in the state assignment. And clearly, the prevalence of hybrids was underestimated if we ignored the observation process. Okay. Nina, Lucille, and Sarah got their work published in peer-reviewed journals. But maybe more importantly, their results have been used to establish a perspective about the long-term viability of the population. And these results are also used by the Ministry of Ecology and the Ministry of Agriculture actually to define quotas for regulation of wolves with the aim to decrease depredations on livestock. And the way our scientific results are used by others is the subject of much discussion in our team. To put it a bit crudely, it is sometimes difficult to accept that the results of your capture-recapture analysis, as fancy as it is, are used to kill animals. Okay, so we're done with the how many questions. What about the where question? So I don't know about your country, but in France, decision makers and politicians love their indicators, which are simple quantities that summarize complex information. And our National Observatory for biodiversity in France releases all sorts of indicators every year. For example, the proportion of the country, here on the y-axis, over time, here on the x-axis, occupied by large carnivores. And on this graph, what you see is the proportion of wolf that occupies the country. And you see that it's been increasing since 2003, and that we have around 5% of the country occupied by wolves in 2017. This proportion of area occupied by the species is what we call occupancy in ecology. But like population size or density, occupancy is difficult to estimate in the field. And let's take an example in which our study area can be divided in, say, 40 cells, out of which 10 are occupied, okay? The ones in yellow here. Then true occupancy is 25%, 10 over 40. Now, for some reasons, you manage to detect the species in only 6 sites out of the 10 sites that are actually occupied, the red cells here, right? Then you obtain a naive estimate for occupancy of 15%, 6 over 40. In other words, occupancy is underestimated if imperfect detection is not accounted for. Again, we will make use of hidden Markov models to infer the fate of these sites by explicitly separating observations from ecological states. And here the states are a species is present or not at a site, and the observations are the species is detected or not at a site. So each site may be in state unoccupied, big U, or occupied, big O, with probability psi1, the probability of occupancy, of initial occupancy. And our Markov model here is driven by the identity matrix, meaning that you stay in the state you are initially in. If you're occupied, you stay occupied, and if you're unoccupied, you stay unoccupied. The observation process, as usual, connects the states, unoccupied and occupied here, to the observation. Here, 0 for non-detected and 1 for species detected at the site. If the site is unoccupied, then the species cannot be detected. Okay, so cannot be detected with cannot be detected, so with probability 1. If the site is occupied, then the species is detected with probability P and missed with probability 1 minus P, right? And to summarize, for single season, also called static models, we only need two parameters, psi1, which is the probability that the site is occupied, and P, the probability that a species is detected. It is then relatively straightforward to extend this model to multiple seasons and to capture colonization and extinction processes or events through probabilities gamma and epsilon, where gamma is the probability that an unoccupied site becomes occupied, and epsilon is the probability that an occupied site becomes unoccupied. The only difference here is in the way we specify the state process here. And we have a Markov chain for the dynamic of the states from time t to t plus 1 in columns, t in rows, and t plus 1 in columns. And this Markov chain is driven by the extinction and colonization process, the gamma and epsilon. For example, the probability of being occupied at t plus 1, yeah, this column, given that you were unoccupied at t, here, this is the first row, is gamma, the colonization probability. Julie Louvrier worked a lot during her PhD on this family of models. She survived her PhD despite spending endless hours waiting for some results coming out of the computer. As an example, I will focus for a few slides on LYNX. The species is monitored in France by collecting all sorts of signs of its presence, like predations, collisions with cars, tracks in the snow, camera traps, or scats and hares that leave behind. The distribution in France is spread over three main populations in the Vosges mountains here, the Jura mountains, and in the Alps. And from the raw data, we see that the Jura and Alps populations are quite steady, whereas the population in the Vosges mountains here, that one, seems to go extinct, right? The main causes of LYNX mortality are collisions with cars and poaching. The cool thing is that our colleagues from the National Biodiversity Agency have set up a network of professional and citizen observers for wolves and lynx. And this network has around 4,000 or 4,500 members that are trained to recognize the signs of presence for both species. And here you have maps of the sampling effort over time for 3 years. The darker, the more observers. And you see that the sampling effort has expanded in time, right? And in space, mainly driven by the expansion of wolves. For LYNX, this is what we get. In red, you have the raw data, just the detections and non-detections made in the field. And in black, the solid line is the proportion of area occupied by LYNX as estimated by occupancy models, dynamic occupancy models. Overall, occupancy is underestimated when detection is not accounted for, right? Exactly like in our toy example from earlier. And there are also some differences in trends here. In recent years, the occupancy analysis seems to detect the decrease in presence due to the extinction of population in the Vosges mountains. This is not the case for the raw counts. Okay, there is no decrease here in the red dots. This is because the trend in these observed counts in red are blurred by the increase in the sampling effort we've seen just before. Hence, the importance to distinguish the ecological process from its observation. And this is made easy and natural by using HMMs. Well, as any statistical model, occupancy models rely on assumptions that need to be fulfilled for safe inference. Independent detections, no unmodeled heterogeneity, no false positives. And today, I will focus on two of them, starting with the assumption of no unmodeled heterogeneity. Again, I will rely heavily on Julie's PhD awesome work. To incorporate heterogeneity, Julie used finite mixtures like we've seen before for capture-recapture data. She considered 4 states, unoccupied in class 1 or class 2, and occupied in class 1 or class 2. And there are two observations, species undetected or species detected, 0 and 1s. Okay? And heterogeneity in the detection is captured by this observation matrix. If you are a site occupied in class 1, then the species may be detected with probability P1 or missed with probability 1 minus P1. And same thing for class 2 with P2. Okay, so like before, we relied on the sampling effort recorded by our colleagues from the National Agency for Biodiversity. And the issue with sampling effort is that it's difficult to measure on the field because often what we get is data from non-professional folks who do not record their efforts. And so our motivation here was to try and assess whether finite mixtures could capture heterogeneity in the detection probability due to spatial variability in the sampling effort, right? And Julie fitted a model with two classes of heterogeneity in the detection probability, say A and B, and then using the Viterbi algorithm, she was able to decode the states and map the distribution of sites in light blue, and sites B, right? And it worked quite well because sites of class B, which also corresponds to high detection probability, were in areas here and here with high sampling effort, which was quite nice. Julie also worked on accounting for false positives due to species misidentification. Here, you may detect the species on the site with no doubt on its identification, and we call that true. But you may not be sure if it's the species you're actually studying, and we call this observation 1. Okay, this is when you have data in which confusions can occur between the species you're studying and another species, like lynx and wildcats, for example. And to model this data, Julie formulated as an HMM a model developed by David Miller and colleagues. First, if the site is unoccupied, then the species should not be detected, okay? The site is unoccupied. But you have false positives in the ones here. So you may actually detect the species with probability P10, which is the probability of false positive detection, right? Now, if the site is occupied, then you detect the species you're interested in with probability P11, the probability of detection, times B, the probability that the detection is classified as unambiguous, so it's B. And you make an uncertain detection here with probability P11 times the probability that the detection is classified as ambiguous, so 1 minus P. Let's say certain, based on solid facts like dead wolves, genetic samples, or observations confirmed by an expert. And we have data with possibly false positive, uncertain data, which can be unverified observations or observations that cannot be verified by an expert like tracks, wild prey remains, or sightings. And uncertain data represents up to 30% of the work data, which is huge. And usually we just get rid of them, which is a big waste. Why? Because these data occur outside the core distribution area, which could inform managers on colonization. And Julie got help from Valentin Lauré, who was a master's student at that time. And together they estimated occupancy by including all data, certain and uncertain. And they mapped the difference in occupancy between all data included against occupancy estimated with certain data only. And they found that when considering uncertain data, we predict a higher probability of wolf presence at the front of colonization. And this has important implications in terms of management to mitigate depredation on livestock. So Julie did the work and published several papers based on her analysis. And her results are directly used in the National Action Plan for LYNX that was made official earlier this year. There is also a national action plan for wolves in which Julie's results are used to inform decision. Okay, I'm almost done. I hope you're not too sleepy, just in case a little soft music to wake you up. So why HMMs? Well, these models allow making a natural distinction between ecological processes and their observation. Why is this useful? Well, I find it very useful to focus my attention on the ecology while properly handling with the observation process. Also, these models are highly modular with complexity broken in smaller problems. Think of the observation process split in detection and assignment in the hybridization capture-recapture example, or in the false positive occupancy example. And last, there is an entire toolbox of useful methods for estimation and inference in HMMs. And in the end, one may wonder whether HMM is a versatile modeling framework. And it appears so, since all sorts of data can be analyzed with the HMM, like spatial capture-recapture data, telemetry data, data on individuals marked in batches, etc. And you can also formulate integrated population models as HMMs. There are also a few perspectives for HMMs. First, HMMs are so flexible that you can model pretty much anything, but we're still lacking methods to evaluate the quality of fit of these models to data. Second, in my talk, I have covered HMMs in discrete time only, and there are extensions to continuous time, which can be much useful when it comes to analyze citizen science data that may occur at any moment in time, I mean opportunistically. And last, HMMs are used a lot in movement ecology to analyze GPS data and infer behavior. And I wonder whether HMMs could be a framework to merge movement ecology, behavioral ecology, and population dynamics. Before I leave you, I'd like to give you a few words of reflexivity and to think for a minute why statistical ecology? And I hope that will be useful to others. Well, for a long time, I had this complex about not doing theoretical statistics or not doing ecology with a fieldwork of mine, and the community in statistical ecology has allowed me to flourish in this role at the interface between both disciplines. And going back and forth between ecological questions, modeling, the imperatives of biodiversity management, I feel like I'm useful, I have some impact. So listen to Françoise Barré-Senoussi, Nobel Prize, who says it much more convincingly than I do.
Keep in mind the real reason why you decide to become a scientist. Is it for your CV? Is it for your personal interest? To be a leader, to become a star somehow in the field of science? If this is the reason, just forget about it. Now, if you want to become a scientist to try to bring something important for the society, for the others, then go on. Because at the end of your career, you will feel good.
Okay, last point I'd like to make is that everything I told you about is from teamwork and the work of our human-animal interactions research group in Montpellier. Science is made by people, and I would be nothing without the students and colleagues who are kind enough to bear with me. But working conditions in academia are deteriorating in an alarming way because of more competition, more precarity. And let's take a minute to read a few words of the abstract of this paper that was recently published on a national assessment of the mental health of UK doctoral researchers. Doctoral researchers, DRs, are an important part of the academic community. And here, by comparing DRs with educated working controls, we show that DRs report significantly greater anxiety and depression, and that this difference is not explained by a higher rate of pre-existing mental health problems. Moreover, most DRs perceive poor mental health as a normal part of the PhD process. Thus, our finding suggests a hazardous impact of PhD study on mental health, with DRs being particularly at risk of developing common mental health problems. I don't have a magic solution to solve the problem, just wise words from Himanshu Gupta, a colleague on Twitter. In a rush of academia, don't forget to enjoy life, family time, good food, outdoor activities, traveling, etc. And if you're thinking to do it all after you finish an experiment, a paper, school, PhD, postdoc, etc., remember this is just one life and time is not coming back.